My name is Olivia Kohler. I'm a senior biochemistry major and biology and math minor at Duquesne University. I've been doing independent undergraduate research for around 3.5 years. I started in my freshman year in the spring. I work in Dr. Seibert's lab, and our lab is focused on gaining a more complete understanding on how enzyme catalysis happens by mimicking intracellular conditions in vitro, which is basically outside of the cell. It has great relevance in the field of like drug design because drugs typically target enzymes, so they need to be modeled very accurately. And enzyme studies in the past have been done in dilute buffer systems, which don't account for a lot of the macromolecules present inside of cells. So we don't know if that's a very accurate measure of how cells actually how enzymes actually function inside the cell. So my project addresses the fact that a lot of common cellular molecules are negatively charged, so like ATP, which is the cell's energy source, and nucleic acids, which code for our DNA, basically, they're very negatively charged and present inside the cell. So I'm looking at the effect of what they're called polyanions on 4 different enzymes. So far in my research, I've found a really interesting distinction between 2 enzymes that are metabolically very similar, malate dehydrogenase and lactate dehydrogenase. They catalyze very similar reactions, so we expected them to behave very similarly in the presence of ATP, but they actually had very distinct effects, or very distinct behavior from each other. Because independent research projects are typically highly specialized, you're conducting experiments and getting results that no one in the world has ever thought to have done or done. It's really, really cool. And I also really like just the bond that you form with your lab group. My lab group is pretty small, so we get a lot of like one-on-one banter, and like we go out to lab group dinner once a semester. We've gone to my graduate student's house to like make dumplings. It's just really nice to have those bonds. So one of the really cool things about conducting research at Duquesne is that you get to present it at conferences. So myself, I have compiled my research into 3 different posters and presented them at local conferences at Duquesne. I've also had the opportunity to go to Indianapolis, Indiana to present at the 2023 ACS National Conference, and that was a really neat experience. I got to present in a really professional setting and also listen to talks from other really distinguished scientists and really broaden my horizon outside of biochemistry, and I got to just strengthen my scientific communication skills.
My name is Haley. I work with Dr. Seaman in the biology department. I'm a senior bio major. Our lab focuses on molecular evolution and genetics. So my project is called The Functional Evolution of KLK2 and KLK3 Proteases in hominoid primates. So I focus on looking at 2 specific genes that have a different pattern of evolution between non-human primates like chimps, gorillas, and gibbons, and then in how it's different than what it looks like in humans, and then I'm looking at the protein function of that gene to determine how it's changed in different primates. So this is a pretty novel experiment, so there's not that much information on the exact thing that I'm studying, so it's pretty cool to actually have like results that nobody else had come to that like conclusion. Some setbacks. Honestly, things just don't work sometimes, and it takes a long time to get like one result, but like it's very exciting when you do. But sometimes it's slow. So last year I worked in the same lab, and I started this project, and I was able to talk about my first year's results at NCUR in Eau Claire, Wisconsin, which was really cool, and I got a lot of funding for that project. And then I was at Duquesne over the summer doing the summer research program that was fully funded, and then I got the Tri-Beta research grant in August of this year, and now I'm going to be able to present my results from that work at the regional conference in April.
Hi, my name is Mackenzie. I'm a junior in the School of Science and Engineering. I'm a biology major with biochemistry and English minors. I started my research career my freshman year here at Duquesne. I started in the Porter lab. I originally just shadowed in his lab and would attend lab meetings, and then sophomore year I took more of a hands-on role researching plastic pollution. With Dr. Porter, I was able to present at several conferences and really grow my research skills on how to be an independent researcher. The summer after that, I was able to do an internship with the Children's Hospital of Pittsburgh, where I researched inborn errors of metabolism and using all the people I met there and the skills in more biomedical research. Now I do research with the Hillman Cancer Center here in Pittsburgh and the University of Pittsburgh Center for Pharmacogenetics. So I do research in pharmacogenetics, which is basically researching how drugs can affect genes and different genetic variations that cause problems within people, and basically treatments for that. I specifically do research in cancer biology with the Hillman Cancer Center. Basically what I research is a variant called PNPLA3 I148M, which is a mouthful, but basically this variant can do one of two things. One is a precursor for non-alcoholic fatty liver disorder, and patients with this variant are more likely to develop this disease. But the context I'm researching it in is for adverse drug reactions. There's a type of leukemia called acute lymphoblastic leukemia, or ALL, and basically the primary treatment for this is asparaginase, but patients with this variant have extreme drug adverse reactions to this chemotherapy. When it's highly successful in children, but in adults it causes a lot of problems with the liver, similar symptoms to non-alcoholic fatty liver disorder, which causes them to have to stop chemotherapy, which is obviously not what we want to do when we're trying to get rid of cancer. We don't want to cause liver toxicity. So basically I research this variant in the context of chemotherapy. In previous labs, I've been able to do more bench work that is like hands-on and biochemical, which is so interesting, and especially with the genetics portion of the labs. I've really enjoyed doing that stuff, but with my new lab, one of the main reasons I chose it is I get to work with mice. And so I have my own murine model that I get to work with, and so it's been so exciting learning how to work with mice, how to treat them, how to just do everything that requires. I had no clue how much work gets put into breeding these mice and getting them shipped in, and then actually treating them and taking them out. So it's been really exciting to learn new techniques involving mice.